Hi, my name is Tony Cho. I'm a sixth-year graduate student in Professor Sungmin Shin's lab. Please allow me to introduce my project that I collaborated with late Professor Ham. An amino acid residue chain as shown here form hydrogen bonds and side chain contacts to reach the folded state. It has been challenging to identify how these interactions contribute to protein chemistry at single amino acid resolution. So we want to quantitatively identify the critical residues that contribute to folding stability. Site-directed mutagenesis methods have become standard tools to investigate the impacts of individual amino acid residues. For specific backbone, amide to ester mutagenesis is employed that applies chemical modification and perturbation to the system. For specific side chain, alanine mutagenesis is widely adopted. These experimental methods are limited because quantitative comparison among different backbone and side chain sites is difficult. For example, mutations on some key side chain make the protein impossible to fold. Then the folding free energy cannot be measured, so the key side chains cannot be compared as to which one is more important. Moreover, using the same set of conformational ensembles for side chain comparison is more reliable. Two different experimental methods on the same mutant have been reported to generate the unfolded state ensembles that differ by 50%. This shows why using the same ensemble is helpful for consistency. In our study, we quantified site-specific thermodynamic stability at the molecular level to extend our understanding of the stabilizing forces in protein folding. The figure shows how much backbone and side chain contribute to folding stability, where blue residues are the key groups. Our model protein is WW domain, the shortest beta sheet protein with 3 beta strands. We performed an all-atom molecular dynamics simulation of WW domain protein with the Amber force field and with explicit water treatment. The folded state simulation was run at 300 Kelvin. We made 6 trajectories that last 1 microsecond each. For the unfolded state, we were more careful in that we carried out the simulated annealing from 600 to 300 Kelvin for the protein to unfold and then rest. Then 2 microsecond simulation was performed for each trajectory, and the last microsecond was used for analysis. The following structural properties were computed: hydrogen bond, side chain contact, secondary structure, and RMSD. Both the structural and thermodynamic analyses were carried out for every 50 picosecond on these free energy components. For these components, we were able to decompose them into backbone and side chain contributions. Finally, we obtained the overall configurational entropy and the folding free energy. In the theoretical part, we start by obtaining the gas phase potential energy E sub u, using the molecular mechanics force field. Its decomposition into residue contributions is carried out using the equation as shown. Here we have intra-residue energy and inter-residue energy. The inter-residue energy is the interaction energy between ith and jth residue. We assign half of this interaction energy to residue I and the remaining half to residue j. For simplicity, we have a figure of 3 atoms. 2 atoms belong to residue 1 and the green one belongs to residue 2, separated by an angle theta. Then the energy associated with theta can be distributed equally to both residues. However, if we distribute the interaction energy term according to the number of atoms in each group, for example, 2/3 of the energy of theta is assigned to residue 1 and 1/3 to residue 2, We can extend this energy decomposition into contributions from the backbone and side chain groups. The equation is provided as shown. We distribute the interaction energy of the bonded terms according to the number of atoms involved, denoted as alpha. We compute the exact atomic decomposition of solvation free energy using 3D reason methods. We used Kovalenko-Hirata closure relation as well. Then we obtain the overall effective energy F by a simple addition. Finally, we obtain the configurational entropy using the variance of effective energy multiplied by thermodynamic beta over 2. Now we show the results of the MD simulation for the folded and unfolded state. Here are the representative structures for each conformational state. The cyan and pink van der Waals spheres are the side chains that form hydrophobic clusters. We used the K-clustering algorithm to select the representative structure. Here we show the structural characteristics in the folded and unfolded states. In the upper table, we show the native structural characteristics involving RMSD, secondary structural contents, and heavy atom contacts of hydrophobic clusters. In the lower table, we see that the number of hydrogen bonds and side chain contacts. We note that in the native structural characteristics, we observe a large difference between the folded and unfolded states. This is not the case with the non-native structural characteristics. We will see how this structural disparity is translated in thermodynamic manner. Now we plotted the effective energy distribution to obtain the configurational entropy. The x-axis is the effective energy centered at the mean value. The y is the probability. The closer the plot fits ideal Gaussian, better the estimation of configurational entropy will be. We see that the folded state trajectories are converged better. We also show the tabulated data of folding free energy and its components. A large negative value for the change in effective energy was obtained and a positive for the configurational entropy, which is good. We obtained a folding free energy of negative 4.9 kcal per mole with 4.9 kcal per mole of standard error. This error stems from the unfolded state configurational entropy, but the error is less than 2% of the unfolded state configurational entropy. Still, the experimental determination of folding free energy using the temperature jump technique was negative 3.4 kcal per mole, which is comparable. Now that we validated our result of free energy with the experimental value, we extend our analysis into backbone and side chains. Finally, we compare the structural characteristics with that of thermodynamic calculation. The x-axis is the residue number, the y-axis is the change in effective energy upon folding. A is a group of residue, C a group of backbone, D a group of side chains. In figure B, we have the hydrogen bond indicated in red and side chain contacts in blue. It is seen that both the hydrogen bond and side chain contacts are simultaneously formed upon folding, especially in the central beta 2 region. In the unfolded state, hydrogen bonds and side chain contacts are formed independently, as previously seen in table number 3. The non-native structure, on the other hand, their simultaneous formation cooperatively stabilizes the folded structure. This shows that the secondary structure is in general not stable by itself and that hydrogen bond strength is amplified when sequestered in a more hydrophobic environment. In conclusion, we validate our result with experimental studies. The folding free energy was found to be negative 4.9 kcal per mole, which is comparable to the experimental finding. Moreover, we compared our determination of folding effective energy to the delta delta G from the experiments that are obtained from mutagenesis. To do so, we use a statistical measure called Spearman's correlation coefficient. This coefficient measures the strength and direction of association between two ranked quantities. The backbone was found to be 0.73 and the side chain 0.76. This result indicates that our study and experimental findings are strongly correlated. On an ending note, I'd like to extend my sincere gratitude to Professor Ham. She was my role model and pushed me to carry out good research. Dr. Song Ho Jung provided me with detailed guidance that I learned so much. Professor Sung Min Shin has given me trust and helped me along my journey as a graduate student. I thank Minu Kim for lots of technical assistance and the rest of my colleagues. This is the reference, and thank you for listening.